Good evening, good morning, good afternoon, wherever you are in the world. Personalised or precision medicine represents the next major leap in modern healthcare. It's become the optimal direction for treating human disease. Hello, I'm David Solomon, the CEO of the Australian Friends of Tel Aviv University. There are 26 Friends associations around the world supporting the university and its work in addressing global issues and finding solutions to human challenges. Tel Aviv University researchers from medicine, life sciences, engineering, and computer science collaboratively work in the field of personalised medicine using diverse approaches and perspectives, with the ultimate goal of developing novel drugs, technologies, and therapies. Over 4 months, we will be highlighting the impact of TAU's exceptional work in this field, with conversations with some of Tel Aviv's greatest minds. And our guest speaker tonight is the first of these. A big thank you to our collaboration partners for tonight, the Harry Perkins Institute for Medical Research, the Australian Jewish News, and J-Wire. And if I could remind everybody, because we will have plenty of time for questions and answers, that there is a Q&A button down the bottom of your screen. Please use that to ask any questions. We will endeavour to get through as many as we can, and hopefully we will have enough time to answer all of them with a bit of luck. I'm now honoured to introduce our moderator for today's webinar, Professor Henry Brodati. Professor Brodati AO is a researcher, clinician, policy advisor, and strong advocate for people with dementia and their carers. At the University of New South Wales in Sydney, he is Scientia Professor of Ageing and Mental Health, co-director of the Centre for Healthy Brain Ageing, and director of the Dementia Centre for Research Collaboration. He has over 500 publications in refereed journals, is senior psychogeriatrician and head of the memory clinic at Prince of Wales Hospital. In 1985, Gordon Parker and he established the Mood Disorders Unit at Prince Henry Hospital, which eventually became the Black Dog Institute. He was previously president of the International Psychogeriatric Association, chairman of Alzheimer's Disease International, and president of Alzheimer's Australia. He's an officer of the Order of Australia and a Ryman Prize winner. It's my pleasure to pass over to Professor Brodati to introduce our guest speaker.
Thank you, David, and thank you for the privilege of being able to introduce our guest speaker, Tal Dvir. Tal brings together knowledge and scientific research from two different fields, from biological sciences and from engineering and material science. And it's this bringing together of different disciplines, which is always important for new discoveries. He is the director of the Laboratory for Tissue Engineering and Regenerating Medicine at Tel Aviv University, where he's a professor in the School of Molecular Biology and Biotech, and also at the Department of Material Sciences and Engineering. He studied in Ben Gurion University and at MIT in Boston. He established his laboratory at TAU in 2011. And he leads a multidisciplinary research team looking at new technologies based on cutting-edge materials and stem cells for engineering and regenerating diseased tissues and organs. It's the way we're all going. Many of you in the audience have got new knees, new hips, new various organs in our body, and this is becoming more and more common. Currently, Tal's laboratory designs and develops smart bio and nanomaterials. Nano is really small, and technologies for engineering complex tissues such as heart, brain, which is my area, I'm really interested in that, spinal cord, intestine, eyes, and more. His lab does 3D printing of tissues and organs. It sounds fantastic. Engineered tissues with built-in electronics and development of personalized engineered tissue implants. We're all going to live forever because of Tal and his work. The research of the lab has great clinical potential, which may make a change in how diseases are treated prolong our lives and improve our quality of life. It's my pleasure to introduce Professor Tal Dvir. Thank you.
Thank you very much, Henry, for the kind introduction. And thank you all for joining us today. So I'll share my screen. Okay, so can you see my screen? Okay, so okay, great, thank you. So my lab at Tel Aviv University is working on tissue engineering and regenerative medicine, and we're engineering different types of tissues from the heart, brain, spinal cord, intestine, kidneys, retina, and other tissues. And I'll give a short overview of the things that we're doing at the lab. So before starting to say what we're doing, I'll explain what is tissue engineering, what is the concept of this technology. So in tissue engineering, we have a patient, as you can see here, with a defected organ here on its left side. And what we do in tissue engineering, we're engineering an equivalent tissue, a substitute tissue in the lab. And then we can take this tissue and remove the defected one and transplant, of course, under sterile conditions, the engineered tissue instead of the defected one to regain function. So this is the concept. And what do we really do in tissue engineering? So we can take cells, preferably we'll take them from the patient because we don't want to provoke an immune response. And initially we cultivate these cells in petri dishes or flasks to expand their number. And then we need to create tissues. And tissues in the body are not just cells. Cells are very important. However, the material in between the cells gives the cells their structure and function. So what we do in the lab, we develop different types of biomaterials. These should be three-dimensional materials, porous materials. We can add, so we see the cells inside these biomaterials. We add different factors, growth factors, small molecules, nanoparticles. And essentially, we're doing everything we can to allow the cells to assemble into a functioning tissue, and then we can take it and transplant it back to the patient. And if we're talking about the heart, this is a slide about the heart, but it can work the same concept on other tissues as well. We use what is called induced pluripotent stem cells. These are cells that used to be regular cells from the body. In our case, fat cells that were taken from a patient. And what we do, we reprogram these cells by genetic engineering to become like embryonic stem cells. And then they have the capacity of becoming any cell type and any tissue type. So in this case, with the heart, we can create cardiac cells or endothelial cells that form blood vessels. And there are a few approaches. One of them is to create thin 2D constructs. The other one is to create real three-dimensional cardiac patches. Or we can even now engineer a whole heart to replace the entire heart. And when I talk about the biomaterials that we're using, there are different forms of materials. We can use hydrogels, where we take the cells and just mix them with the biomaterials with these hydrogels, and then we can inject or transplant these tissues. We can have preformed scaffolds, some kind of sponges where we seed the cells into these pores, and afterwards we transplant these tissues. Or we can create fibrous materials that really mimic the extracellular matrix, the material that we have in the body, such as the collagen fibers. So it mimics the morphology of these fibers. And when we want to create cardiac patches, for example, and we take into consideration different things like the mechanical properties of the biomaterial, the morphology, topography of the material, biochemical content of the material. If we take everything into consideration, we can create such cardiac patch. This is a few centimeters in diameter. a few millimeters in thickness, and it contracts nicely in the lab for a long period of time. We can take these patches and transplant them, and if we do that in animals, we can see a very nice regeneration after 1 month of the infarcted hearts that had the heart attack. And if we wait enough time, we can see a very nice rejection of these cardiac patches, because usually the biomaterials that are used are either synthetic or natural, but they're not really fitting the patient's body immunological profile. So what we've decided to do in the lab is to create a completely autologous, meaning from the patient's tissue. So what we do, we take fatty tissue from the patient, we isolate the cells from this fatty tissue, and we reprogram them, as I mentioned, with genetic engineering to become embryonic-like stem cells. And the biomaterial in between the cells is processed to become a scaffolding material, which is autologous, meaning comes from the patient itself. And once we put the cells inside these biomaterials, we can create any tissue type that we wish. And this will not provoke an immune response because it comes from the patient. So we can create, this is how the biomaterial looks, and we can create cardiac tissues with the same morphology of the real cardiac tissue. We can create blood vessels within, and these patches, they can contract nicely in the lab for a long period of time. And when we take these patches and transplant them in animals to evaluate the immune response, we can see that these autologous materials, they provoke less immune response, almost none. And this is extremely important when we want to regenerate any type of tissue, because we don't want it to work for a limited period of time. We want it to work for life. So now we can take these cardiac patches and transplant them on the heart, as you can see here. This is the patch, this is the heart. This animal had a heart attack, and we were trying to regenerate the heart. So from this movie, you can see two things. One of the things is that there is an open chest surgery for this treatment. We need to open the chest and transplant with sutures these patches. So it's not ideal to open the patient after the patient had the heart attack. This is one thing. Another thing that you can see is the 2 stitches here to secure the patches, and I'll talk about these stitches later on. So to avoid this open chest surgery, this is in the case of the heart, but it's the same thing with the brain, spinal cord, and any other tissue. What we decided to do is to create a hydrogel from the patient's own material. So we take the patient's own material, we mill them in the lab, we process them to become liquid in room temperature. Now we can mix the cells here at this stage, and in 37 degrees, meaning the body's temperature, this solidifies. So we can inject by a catheter these tissues wherever we want to the body, and they will form the tissue in the body without the need for surgery. So based on this technology, first of all, a disclosure, we formed the Matrixelf platform technology. The company is already in Tel Aviv booth, exchange. And what we do, we take this fatty tissue, we isolate the cells, we reprogram them to become embryonic-like stem cells. The extracellular matrix is processed to become a personalized hydrogel. And then when we mix the cells, these stem cells, which have the capability of becoming any tissue type, we can create different types of tissues. This is a partial list of the things that we've done at the lab. For example, we've created adipogenic implants for reconstructive surgeries, motor neuron implants for spinal cord injuries, cortical implants for brain trauma, dopaminergic implants for Parkinson's disease, and cardiovascular implants for cardiovascular diseases. And we can even use more advanced technologies such as 3D printing and microfluidics to create more complex tissues. So I'll give some examples. So here you can see the cardiac implants. 2 weeks before, they used to be stem cells, and a month before, they used to be fat cells. So now they became cardiomyocytes, cardiac muscle cells, which are functional, as you can see here. And they, once we create these tissues, they contract and transfer the electrical signal, and we can inject them to, in this case, to mice hearts. And we're showing that we're elevating the ejection fraction, meaning that the heart is becoming more functional when it is treated. We also did it with bigger animals, with the pigs. As you can see here, we're injecting it directly to the left ventricle, and we're actually regenerating pig's heart after myocardial infarction. And this is a prerequisite step before reaching clinical trials. So I mentioned that we can 3D print tissues. So what we do here, we look at the patient's left ventricle, the CT image of the left ventricle. This is it. And what we do, we isolate the blood vessels. We transfer these blood vessels to the computer. And then we can create cardiac patches according to the anatomy of the patient. And before we do that, what we do, we have some mathematical modeling to bring in more blood vessels into the patch to better nourish the engineered tissue. And then we go to the 3D printer that we have in the lab, and we're printing patients' own materials and patients' own cells, cardiac cells for the cardiac tissue, and endothelial cells and smooth muscle cells for the blood vessels. And this is how it looks. And again, it's according to the patient's anatomy, biochemical, and cellular properties. So there will not be an immune response, and there will not be a rejection after a transplantation. And eventually, we're getting this patch. So it is filled with the blood vessels and cardiomyocytes. If we look at the cross-section of one blood vessel, you can see that it is intact. And it is functional. This is the cardiac tissue. This is the blood vessel, for example. And you can see the transfer of the electrical signal and the contraction of these cardiac patches, meaning that these are functional patches. So not only that we can use these to regenerate tissues, what we can do, we can, for example, inject drugs, different types of drugs to these heart patches, and then evaluate how a certain patient or how certain drugs affect a human heart, because these cells are came from a human patient. So we can evaluate, and this is in collaboration with the Bayer company, so we can evaluate how different drugs affect the heart. And this is not done in Petri dishes. This is done on 3-dimensional cardiac tissues with blood vessels. We can also use these patches, and when we manipulate them a little bit, and I remind you that everything is fully personalized, when we manipulate them a little bit, we can create injectable vascularized heart patches that open heart after injection, and they will stick to the heart. So when we had this success with the 3D printing of these cardiac patches, we wanted to move on to print more volumetric tissues. So here you can see that we print, again, from the patient's own materials and patient's own cells, really nice structure, volumetric structure, but this is not really interesting. It's nice, and it has some challenges. In terms of printing, we had to invent new technologies for 3D printing, but we moved on, and in 2019, we printed for the first time a human heart with the blood vessels. This is a tiny heart. rat-sized heart. You can see it here in the middle of the printing. These are the ventricles. This is the blood vessel. And you can see here the end product. And these hearts are contracting, or the cells are contracting. But initially, they couldn't contract synchronously, and we had to teach them how to do that. And we did that by two ways. One of the approaches was to mature these hearts in specialized bioreactors, where the hearts were hung on here, and they were circulated with the culture medium with all the growth factors and nutrients. And while we circulated these hearts with the medium, we stimulated them. You can see here the stimulator. And this taught the cells how to actually speak with each other on the electrical level. Another approach was to take these hearts and use the body as a bioreactor to transplant them, these hearts, in parallel to the native heart, and have the blood nourish these hearts. And after a while, we see that they're contracting. Eventually, when we isolate them, we could see that they're contracting, and you can see here the signal transfer from the right atrium to the septum, and then to the ventricles. And this is how heart should work. So we accomplished that. We can also print, so if we can print the heart, of course, we can print parts of it. So here we printed the atria. This is how it looks on the printer. This is the end product. And you can, I hope that you can see here that it contracts nicely. And what we can do with that is evaluate how, you know, we can have conduction disorders, and then we can add drugs and see how these drugs affect the engineered agent. So this is about the heart. We'll get back to the heart soon. Another thing that we do is we create spinal cord implants by the same approach. We take this fatty tissue, we create these embryonic-like stem cells. The ECM is processed to a personalized hydrogel, but then when we take the stem cells and put them in the hydrogel, we don't mimic the embryonic development of the heart. In this case, we develop the embryonic development of the spinal cord, and we create spinal cord implants to treat paralysis. So this is how it looks. 30 days before, these tissues were just stem cells, and 2 months before they were fat cells. Now they're, you can see 3D neuronal networks. And when we inject them instead of the injured spinal cord in a chronic model of this injury, this is the MRI of the animals. You can see that the implants fully regenerated the injured spinal cord, compared, of course, to injecting just cells or untreating these animals, or just putting hydrogel. So here you can see a very nice regeneration of the spinal cord. And when we look at the locomotion of these animals, so first of all, this is the untreated animal walking on a machine that is called the catwalk. And this catwalk captures the ability of the animals to put pressure on the legs. So this is leg 1, 2, 3, and this is the 4th one. And you can see that one of the legs is not really operating. It pulls the leg because there is an injury. And if we look at the full implant, you can see that the animals walk like that, and this is extremely nice. And based on that, we formed Matrisoft, which the company that our goal is to be in clinical trials in 2.5 years. We already started speaking with the FDA, getting their guidance towards these clinical trials. But sometimes we do not need to engineer whole tissue. Sometimes we just need a few cells to be injected. For example, in Parkinson's disease, we don't need the full patch. But if we just take the cells and inject them, most of them will die because they cannot form any interactions with their surroundings, and they die. So what we've decided to do was to encapsulate single cells or a few cells inside the hydrogel, the personalized hydrogel, and we did it by a microfluidic device that we developed in the lab. So here we're injecting the hydrogel with the cells. Here we're injecting oil, so we create tiny emulsions. We heat it up to 37 degrees. Now these droplets are solidifying, and we can take them and differentiate them to the different types of cells. In this case, to dopaminergic implants or dopaminergic cells, as you can see here. We cultivate these cells in the lab for a long period of time, for 90 days, and then we can evaluate that they're releasing dopamine, for example, and we're now evaluating their ability to regenerate Parkinsonian brains. Another thing that we can do is differentiate the cells inside the hydrogel to cortical implants for brain trauma. We can create retinal cells for age-related macular degeneration, and already injected them to the back of the eye to regain the function or to stop the deterioration of this retina. So now we're back to the heart for a few slides. Another issue that we have with the hearts is the transfer of the electrical signal inside the biomaterials. The biomaterials are not really conducting, and they may prevent the signal transfer. So if we see the cells inside these pores, you can see them here, and these cells can contract, and this is the pore wall. They're not really speaking with each other on the electrical level. And what we've decided to do is to incorporate gold nanowires into these biomaterials so that the cells can interact with each other. So this is interaction without gold nanoparticles. This is with gold nanoparticles, and everything is contracting nicely. This is the patient's own materials without gold, 4 nanometer gold particles or 10 nanometer particles. And you can see that everything is contracting nicely when we have gold nanoparticles. And what we can do with that, we can, first of all, image the treatment. So we can inject it to the heart, and we can see where is the scar tissue in the heart and where is our treatment. And if it doesn't cover everything, we can re-inject these hydrogels with the cells and gold nanoparticles. And eventually, we're showing that we can regenerate the infarcted heart by injecting these gold nanoparticle and ECM or these hydrogels with the cells from the patient. So it works nicely with the cardiac cells that are electrogenic cells, meaning that they transfer electrical signal. We wanted to evaluate whether it works nice as well with the neurons, which also transfer electrical signal. And this is what we've done. We created these gold nanoparticles on the fibers of the hydrogel. And here you can see without gold, the neurons are not really developing fast. With the gold nanoparticles, we have nice neurites and long neurites. And this is extremely important when we want to regenerate the brain or the spinal cord. So what we see here that gold nanoparticles, which are approved by the FDA for different treatments, and are inert to the body, they are biocompatible. They can improve the function of engineered tissues. So if you remember, I've shown a movie with the 2 stitches on the heart, and I said that it's not ideal to have stitches on the heart, especially to patients just had a heart attack. So what we want to do next is to exploit the properties of gold nanoparticles. They have really remarkable properties that if we illuminate them, irradiate them with near IR light, they convert this energy to a thermal energy. And what we were able to do is to fuse these cardiac patches to the heart just by illuminating these heart patches with the laser. And now we do not need any sutures, because these heart patches are secured to the heart without any stitches. So you can see it here. This is after 30 seconds of illumination, and we can see nice attachment. You can see it also by electron microscope. This is the heart and this is the patch, and it sticks nicely. Now, the last series of projects that I'm going to talk about is what we call cyborg tissues, integration of electronics with engineered tissues, not just because it's fun and cool to engineer electronics with living systems, because initially we wanted to evaluate the ability of our tissue to propagate the electrical signal and to contract, to be able to monitor that. So what we've done, we created meshes of nanoelectronics. These were porous electronics. We incorporated them with the biomaterials, and eventually we had scaffolding materials, 3D scaffolding materials with many, many nanosensors within. So we cultured cardiac cells, and immediately we could see on the computer the function of the tissue. This is just one electrode, but we had many, many electrodes within. So we could see the contraction of the cells. We could add drugs and immediately see how these drugs affect the tissue. We could see it on our computer. So it was a really nice project, a very good publication in Nature, very nice patent for us, but it wasn't enough. Because in this case, we could see the function of the patch, but we couldn't see, but what if the patch was not working properly? We couldn't do anything. So the next step was to create the next generation of these cardiac patches. And in addition to the sensing ability, we want to have the ability to type on the computer and provide electrical stimulation, for example, as a pacemaker, or to type on the computer and release different types of drugs. So if we think about the technology, the patient is sitting in his house, he's not feeling well. Obviously, there's something wrong with the patient's heart, and the physician can log onto the computer, evaluate the function of the heart, and from afar, decide how he wants to activate it. If it senses that there are not enough beats per minute, can type on the computer and provide electrical stimulation. If it senses that there is not enough oxygen in the left ventricle, can type on the computer, release growth factors that promote vascularization, the formation of new blood vessels. Or if it senses there is inflammation, release anti-inflammatory drugs. And these are not things that I imagine. These are things that we actually did in this Nature paper. We then moved to create more stretchable electronics that can release different types of drugs in parallel. We can now release up to 20 different drugs, and only when we want to release them, meaning that they sit in a depot on the electronics, and when we decide, we can type on the computer and then we can start releasing them. And then I was asked, what happens if the drugs are ending eventually? So what we've done next was to develop an approach to reload drugs just by injecting the drugs into the bloodstream. They will not act on any tissue. They will circulate and find the biomaterial, the electronics, stick to it, and then it will be released over and over again. So there is no need to be afraid that the drug is ending. Other things that we can do with the electronics and engineered tissues, so we can print them together. So we developed a formulation for electrodes, soft electrodes and flexible electrodes. And we're now printing the patient's own materials, the patient's own cells, and the electronics together, as you can see here. And this is extremely soft, as you can see in this movie, and extremely flexible, as you can see from this movie. And eventually it can do the same things that we got from the different approach where we created the electronics by lithography. We can do it now with 3D printing and have the same recording ability and stimulation. And the last slide that I'm going to show is a new project. It's not so new, but it still was not published, but we can now create a whole heart, and the entire heart is filled with electronics on strategic areas. For example, on the left ventricle, we have electronics that can release angiogenic factors. We can have on the septum electronics that will activate the heart if there's conduction disorders. And again, everything, the electronics is placed everywhere inside this engineered heart. And with that, I would like to end up, acknowledge my group members, collaborators, and my funding, and thank you for listening.
Well, thank you very much, Professor Dvir. This is just amazing work. It's almost science fiction, you think. And, you know, I've got 100 questions of my own, but we're just looking for more questions in the Q&A. So please, audience, please put in your questions and I will put them to the professor. So it's, maybe I could ask you, Tal, in your dreams, where do you see the future? What is your ambition? Where would you like to be your legacy?
So you know, I'm not talking about my research. I'm talking in general on regenerative medicine. In yes. Regenerative medicine, what I think will happen, and this is a dream that I'm sure that will come true, is that in 10, 15 years, we will have printers in every hospital, and we will print organs to treat patients. And these are things that I truly believe that will happen. It will take a while. We'll start with simpler tissues like skin and cartilage, and then we'll move on to bone and other tissue, and eventually we'll reach heart, kidney, retina, and eventually brain.
Well, I'm really ready for the brain. Now, it just occurs to me that there are a lot of issues here. There are ethical issues, there are health service issues, the costs, there are adverse events we haven't spoken about, maybe they do occur. There's the philosophical issue about people living forever. You know, there's several things there. I'm sure you've had discussions about them. Would you like to just elaborate? And I'll look at the Q&A.
So I'll be rude and I'll say that I'm not dealing with ethics. You know, if people, we provide the technologies, and eventually, you know, if people would like to use it, they will use it. I think that I cannot say anything about the ethics. About at the cost of these tissues, I'm sure that the patient that will need a new spinal cord, the insurance company will pay. It will not be, I'm sure it will not be that expensive eventually. You know, everything costs a lot at the beginning, and then you know, the cost is reduced dramatically. So you know, we're trying to provide better lives to people, and eventually, you know what they say, that the person that will live to 2,000 years, it was already born. I truly believe that it is true. I don't know if we live here on Mars or anywhere, but we try to develop the technologies to do that.
Thank you. One of the questions in the chat, in the Q&A, is how can you be sure that the nerve cell with a transected spine connects up to the right nerve? And, you know, you want to move your right arm, your left leg it goes forward instead.
So this is why we're doing the experiments. And in animal experiments, we see that actually these mice are working properly. What we all know, and I'm sure that you know, that the body is plastic. Our body is plastic, meaning that you help it to regenerate itself. So so it's not that these tracts will be integrated with other tracts. There will be adjustments in the body. This is what I believe that is happening, that the new tracts are now activating, or the engineered tissue knows how to find the right tract.
So are there any adverse effects in this work that you found in humans or in animals?
We couldn't see anything. You know, I'm often asked, but did the animal, what did the animal feel? You know, I cannot answer that. Perhaps it feels pain, but what we do see is that these animals that were treated with the implants regain weight, meaning that they feel fine. You know, when we reach clinical trials, we know exactly if we got sensory as well as motor ability.
And so are there limits to this? I mean, one of the questions is about pancreas, for example. Are there limits to which tissues we can look at regenerating?
So there used to be limits when we used regular stem cells, adult stem cells, because these cells were not able to become neurons or cardiac cells or liver. But now when they're the induced pluripotent stem cells, the genetic engineering that reprogram the cells, somatic cells, normal cells from the body to become embryonic-like stem cells, we can create any tissue type that we wish.
So you saw a future where every hospital will have their printer and be able to print the tissues that we need. What are the fundamental problems that need to be overcome to get to that stage?
So first of all,
printing organs for patients, yeah.
Yeah, so there's first of all these hearts, for example, that we engineer. If we're talking about cardiac patches, I think that we're close to that. There are a few regulations that we need to overcome, but with patches, we're okay. With the full hearts, we need more blood vessels within these hearts. We need a lot of cells to create a big heart, and we need to know how to mature these hearts to have ejection fraction as a normal heart, because now it's still premature. And this is what we're working now on, to mature these hearts to have a nice pumping ability. We're still not there, but I'm sure that it will happen.
And what do you think the timeline is for human trials to take place?
So I believe and hope that in 10, 15 years, we'll be able to have hearts that are working properly in the lab.
10 or 15 years, okay. I'm just thinking about my age, whether I'll make it or not. All right. I mean, the cyborg tissues, where somebody remotely can control this. You know, I'm thinking about Putin in Russia, working with people and putting this stuff into their bodies. And if they don't succeed in what he wants them to do, he can kill them or make them do other things. You know, it has all this sort of ramifications of the sci-fi world. Any thoughts about that?
Well, so we're not working on, cyber technology. However, Tel Aviv University is famous in, we have one of the best cyber technology here.
Yeah.
And you know, it's like hacking hacking the car after there would be autonomous cars. There would have to be cyber security everywhere.
Yeah. I mean, we know now with drones that somebody in Washington can kill somebody anywhere else in the world with tracking their drones, and this really opens up a whole new world. I just wonder if any more questions from the audience. I think I've covered all the ones that are in our room, in the Q&A room there. And we want to leave some time for David to tell us a bit more about Tel Aviv University. the part about gold I thought was quite fascinating, that you can use, you can have it in the body and only be activated by using light, laser lights. And I know in Alzheimer's, which is my area, they're using ultrasound to activate molecules as well. Has that been happening in your world?
So we can activate with near IR light, we can activate with other triggers as well. Here, what we've done for the integration of the engineered tissue to the heart without the sutures, relied on their physical properties, which they convert energy, the light energy, to thermal energy. But people are using gold nanoparticles for imaging and for killing cells and for activating cells. These are things that are already that were already done.
Yeah. So for those of us who are overweight, I'm not one, unfortunately, because it looks like our fat could be our new heart, and that's an amazing thing. There's a very specific question here. Do you think you could generate a more reliable sinoatrial mode than what nature has given us?
Well, this is a very interesting question. We're working on bionic organs as well. So what we do, I haven't spoken about it. We can create the conduction system in the lab now. We have a student that is working on that. And we can basically decide how we want to activate the heart, or where to put this the conduction system, the cells that can transfer the electrical signal in the heart. We can decide where we want to put them, so we can act, or we can dictate the pattern of conduction. We need to think later on what would be a better working heart, how to what's the pattern of the electrical signal, but we can print them wherever we want inside the heart.
Okay. I think we've run out of questions, Prof. So I will hand over to David to. Oh, I see. There's another 10 minutes left. But David, I don't think we should just prolong it for the sake of it, so I'll hand it over to you.
Fantastic. Thank you very much, Henry. Tal, if I might, you're obviously got a very, very broad scope in terms of your laboratory, the work that you do. Are there any areas that you haven't told us about tonight that you think might be interesting to the audience?
Well, we're working on printing kidneys, for example. We're working on 3D printing of the retina with different layers, the epithelial layer, the endothelial cells for the blood vessels, and the photoreceptors. We're creating electronics-based intestine. So we have the peristalsis through the electronics, so we can move this tissue through the electronics. And there are so many other things that we do in the lab, but yeah, basically these are the areas in tissue engineering and regenerative medicine.
Fantastic. And we talked before about a variety of disciplines, the multidisciplinary nature of your laboratory. Could I ask you to explain how does that actually work? How do you bring people from different disciplines, and what are those disciplines? And how do you get them to combine to create such incredible results?
So yeah, first of all, we have a big lab at Tel Aviv University, and the students or postdocs that I have are from different fields. So we have chemists, we have biologists, we have engineers, material scientists, electrical engineers, medical doctors. So the lab is really multidisciplinary. However, it's not that we each of these person is doing only the things that he knows what to do. when I have a new student, I always tell them, well, you need to gain knowledge, multidisciplinary knowledge. So each student will have its own project, and it will deal with chemistry, material science, and everything. And you will learn everything from the other guys in the lab. So it's not that he will collaborate and they will do their part and he will do other parts. The students are doing everything, and this is how they learn how to be to do interdisciplinary science.
And Tal, are there other teams in Israel that you're working with as well?
So we collaborate a lot with the clinicians because we need to transplant these tissues, and we want to recheck the clinic. So we have a collaboration with the Ichilov Hospital in Tel Aviv, working on the engineered retina. We have a collaboration with the Tel HaShomer Hospital on the kidney. We collaborate a lot when I need other things, more advanced chemistry, for example. I have a few friends here at the School of Chemistry at Tel Aviv University that they always have a solution for any problem that I have.
And your funding, tell us about how you get funded to do all this fantastic work. By the way, the research that Tal describes, the journals, I looked them up while he was talking, they're very high impact factor journals. This is top-notch, world-breaking research. Yes.
Thank you, Henry. So the funding, either from Israeli foundations, however, we're limited in Israel. We have European foundations, the ELC, what is called the ELC. Donations, of course, we have some of these. As I mentioned, we have a really big lab, so we need a lot of money. And it's a very expensive research, working with these stem cells and these experiments and these transplantations. Knock on wood, we're doing fine.
And the research funding, in my experience, doesn't cover the cost of research, doesn't cover your infrastructure and associated costs. And so it's really important to get this extra funding, such as the Friends perhaps going to try and raise. Yeah.
Exactly. Yeah. So for equipment, for example, we cannot buy new equipment from grants, from foundations. They're not allowing us. So this is why we have donations, so we can buy new equipment every few years to cope up with the cutting edge technologies.
Yes. David, back to you.
There is another question that's come in from the audience, a little bit specific, but nonetheless, let's see what we can do to answer it. Are you able to elaborate on how the patch on the heart can help with cardiomyopathy?
Well, what we when we have conduction disorders, or when we have areas in the heart that are not transferring the electrical signal, we put the patch on the heart, and then we saw that already after 2 days there's an integration between the healthy part of the heart and the patch. And we're bridging these sick or diseased areas with these patches, like what we do with, I don't know, with our pants when we need to put a patch or any other thing. So we do it with the heart, and it works.
Tal, I'll declare personal interest in this next question. I have a son who was diagnosed with type 1 diabetes when he was quite young, and I'm constantly scouring the world looking for solutions to that problem. And I'm wondering whether there is the potential to use this technology to recreate a pancreas that will actually produce the required levels of insulin.
Yeah, so people are doing that. My mentor at MIT is working on that as well. They formed a company. They now show that they can create small patches of islets of insulin-secreting cells. And the main issue with the diabetes is the immune response. You cannot use, it's not like you're replacing the cells, the body with the better cells, because if you take the cells from the patient, again, it's an autoimmune disease, and it will attack it. The challenges over there is how to create biomaterials that will cover the cells, will not allow the immune system to penetrate into and kill the cells or attack the cells, but will provide the ability to secrete insulin outside. And then they have really nice results with that. I think they formed a company now that they're looking at clinical trials already.
That's amazing news. As Henry said previously, it's the world of science fiction that's actually coming to fruition in the here and now. Look, I would just like to say a massive thank you on everybody's behalf to Professor Tal Dvir for what I think you will all agree was a very thought-provoking session. Also, our sincere gratitude to Professor Henry Brodati for his expert moderation. And thank you again to our collaborators tonight, the Harry Perkins Institute for Medical Research, the Australian Jewish News, and JWire. As I mentioned at the outset, this was the first in a series of 4 webinars highlighting the impact of work in the field of personalised medicine being done by TAU's exceptional researchers. In April, we will be examining genomic approaches in personalised medicine with Professor Karen Avraham. In May, the topic will be the role of AI and big data in personalised medicine with Professor Noam Shomron. And in June, we'll finish with a real highlight looking at revolutionary therapeutics utilising personalised medicine to treat cancer, featuring Professor Dan Peir. Now, if you're listening live or you registered for the first webinar, we will automatically send you details of the forthcoming sessions. If you happen to be listening to a recording, please go to aftau.asn.au. So it's aftau.asn.au and just follow the links to register. Thank you all for attending, and we look forward to seeing you all again soon. Bye for now.